Decoding the cellular symphony. The crucial role of ribosomes in cellular biology. 1. Introduction. Molecular maestros. Within the microscopic orchestra of cellular biology, ribosomes stand as indispensable conductors, orchestrating the synthesis of proteins, a fundamental process for the sustenance and functioning of all living cells. 2. Definition and structure of ribosomes. Molecular factories. Ribosomes are cellular structures responsible for protein synthesis, translating the genetic code from messenger RNA, mRNA, into functional proteins. These molecular factories are found in the cytoplasm of cells and, in eukaryotic cells, also within the endoplasmic reticulum. Dual subunit structure. Ribosomes possess a distinctive dual subunit structure, comprising a large subunit and a smaller subunit. The subunits work collaboratively during protein synthesis. 3. mRNA translation. The blueprint for proteins. Genetic code deciphered. The process begins with mRNA carrying the genetic instructions from the DNA. Ribosomes decode this information, translating it into the language of amino acids, a crucial step in protein synthesis. tRNA. Molecular couriers. Transfer RNA, tRNA. Molecules ferry amino acids to the ribosome, aligning them in the correct sequence according to the mRNA code. 4. Ribosomal RNA, rRNA, the structural backbone. Architectural foundations. The core of ribosomes is composed of ribosomal RNA, rRNA. rRNA serves as the structural backbone, providing the necessary scaffold for protein synthesis. Catalytic roles. Certain regions of rRNA also exhibit catalytic properties, participating directly in the chemical reactions that link amino acids together to form a growing polypeptide chain. 5. Protein synthesis stages. A choreographed ballet. Initiation. The ribosomal subunits assemble around the mRNA with the help of initiation factors. This marks the beginning of the protein synthesis process. Elongation. Amino acids are added one by one to the growing polypeptide chain. The ribosome moves along the mRNA, reading the code and facilitating the addition of the corresponding amino acids. Termination. The process concludes when the ribosome encounters a stop codon on the mRNA. This signals the release of the synthesized protein, which may undergo further modifications for functionality. 6. Eukaryotic versus prokaryotic ribosomes. Unified function, distinct structures. While the fundamental function of ribosomes is conserved across all domains of life, there are differences between eukaryotic and prokaryotic ribosomes in terms of size and specific components. These distinctions provide targets for antibiotics that selectively inhibit bacterial ribosomes. Target for antibiotics. Antibiotics like erythromycin and tetracycline exploit the differences in bacterial ribosomes, inhibiting their function without affecting eukaryotic ribosomes. 7. Ribosomes and cellular regulation. Regulatory proteins. Ribosomal proteins and associated factors play a role in regulating the translation process. These regulatory elements contribute to the fine-tuning of protein synthesis in response to cellular needs. Stress responses. Cells can modulate ribosomal activity in response to stress conditions, redirecting resources based on environmental cues. This adaptability reflects the dynamic nature of cellular processes. 8. Ribosomes in the endoplasmic reticulum, Destination In eukaryotic cells, some ribosomes are bound to the endoplasmic reticulum, imparting a studded appearance. These ribosomes synthesize proteins destined for secretion, membrane insertion, or incorporation into lysosomes. Secreting proteins. Ribosomes on the ear are integral to the synthesis of proteins that will be secreted from the cell or inserted into cellular membranes. 9. Disease implications. Ribosomopathies. Disorders associated with ribosome dysfunction, known as ribosomopathies, can have severe consequences. These conditions highlight the critical role of ribosomes in cellular health. Cancer connection. Aberrant ribosomal function is implicated in certain cancers. Targeting ribosomes has become a focus in cancer therapeutics, aiming to disrupt the uncontrolled protein synthesis characteristic of cancer cells. 10. Conclusion. The cellular symphony continues. Harmony of life. In the intricate symphony of cellular biology, ribosomes take center stage, translating the genetic code into the functional proteins that define life. From the initiation of protein synthesis to the delicate ballet of elongation and termination, ribosomes conduct this intricate process with precision and finesse. Beyond blueprint. As we unravel the mysteries of ribosomal function, we uncover not only the blueprint of life encoded in our DNA but also the dynamic interplay of cellular components. The ribosome, a molecular maestro in the cellular orchestra, reminds us that the beauty of life lies not just in the instructions written in our genes but in the orchestration of these instructions into the exquisite melody of living systems.